Okay, great. Thank you so much for the introduction and for inviting me here. My name is Ekin, and today I'll be talking about reverse perfusion imaging of the cerebral venous system with DiSpect. So the cerebral venous system is studied much less frequently compared to the arterial system. One reason for this is that the veins show more person-to-person variation as compared to arteries, and also the success of current non-contrast enhanced methods to study venous flow is limited. So arterial spin labeling is not very suitable due to the lack of a single labeling plane with veins, and phase contrast has limited sensitivity to slower flows, and so it's mainly only able to capture flow in major vessels. And our aim with this work is to develop a technique which can measure the draining of the veins, starting from smaller capillaries into larger vessels. So here's just a teaser slide showing the capabilities of our technique to perform venous imaging by tracing the blood flow into the superior sagittal sinus. So unlike phase contrast, which measures velocity, the technique that we will use today measures displacement. So here I want to just talk about the difference between velocity imaging and displacement imaging on a schematic of a tree of vessels. So a velocity measurement corresponds to the particular direction that the spins are traveling to during imaging. And the spins might not continue traveling at this direction unless they're moving at a constant velocity. And on the other hand, with displacement imaging, the spins are initially tagged, and after a particular time, imaging is performed, and the positions from which spins came from into the imaging voxel can be traced. So our method is based on another technique which performs displacement imaging, and this technique is called DENSE. So DENSE uses a spam tagging module, and after a particular time, an image is acquired, and the phase of this image is proportional to the displacement that has occurred during the mixing time. So this technique is only able to capture a single displacement value per voxel, so it assumes no partial volume effects, similar to phase contrast. And for this reason, it's mainly used to study bulk displacement, for example, to measure cardiac strain maps. So inspired by Fourier velocity imaging, we introduced displacement spectrum imaging, or DiSpect, which is a Fourier encoding variant of DENSE. So by varying these displacement encoding gradients, as shown here, instead of measuring a single displacement value per voxel, we can actually measure an up to 3-dimensional displacement spectrum for each of our image voxels. And actually, by capturing a displacement spectrum for each voxel, we can resolve partial volume effects. For example, when spins come from multiple different sources into the same image voxel. Here's just an overview of our data acquisition process. With each of our acquisitions, we acquire a 2D K-space, and we can perform just regular image reconstruction to obtain an image from this K-space. So now each voxel in our image for this particular case will have a 2-dimensional displacement spectrum, and our displacement spectrum is 2-dimensional because we're varying these displacement encoding gradients shown here along 2 separate axes. So here we're showing the displacement encoded K-space, which is actually just the K-space of our displacement spectrum. Each time we vary these displacement encoding gradients and make a new acquisition, this corresponds to a new point in our displacement encoded K-space. Then we can vary these gradients and make new acquisitions to fill up this entire space as shown here. And then the FOV of our displacement spectrum and the particular resolution of our displacement will be determined by regular Nyquist criterion. So it will be the resolution will depend on the maximum extent of the displacement encoded K-space sampled, and then the FOV on the distance between the samples. So here are some more details of the particular pulse sequence we use for our venous imaging studies. We added spatial saturation bands before and after tagging in order to suppress our signal from static tissue. And we performed multiple acquisitions after our tagging, and this effectively samples the displacement spectra at multiple different mixing times. And each of our acquisitions was just a single shot spiral readout. In order to demonstrate our technique, we performed a set of 2 experiments. So the first one, we performed non-selective tagging. And then over a period of 2 seconds, the blood will drain from the capillaries into the superior sagittal sinus. We place our imaging slice right here on the superior sagittal sinus. Our imaging slice is very close to the surface and close to receiver coils, and through this, we can obtain higher SNR. So here are some more details for the first experiment. We image a 2-dimensional coronal imaging slice, and we place a saturation band over our imaging slice in order to suppress signal from static tissue. The displacement encoding directions are shown, resulting in a 2-dimensional sagittal displacement spectrum for each of our image voxels. And we also acquire our displacement spectra at multiple different mixing times. And effectively, this creates a 5-dimensional data set. So we have 2 image dimensions, 2 displacement dimensions, and finally a time dimension. The scan time for this particular sequence was 30 minutes. So here's a sagittal projection of our 3D venogram just as reference. And here's our coronal imaging slice. We select an ROI that's on the superior sagittal sinus from our imaging slice, and then we combine the displacement spectra of all of the image voxels that are in this selected ROI. So then we overlay the displacement spectra, the combined displacement spectra on our sagittal venogram and display the results across multiple mixing times, which results in the following. And you can see that at longer mixing times, the signal becomes more dispersed, and we believe this could correspond to the draining from the capillaries into the superior veins. So however, the information that we can resolve with just a 2-dimensional displacement spectrum is limited. So for example, if we acquire a 2-dimensional coronal displacement spectrum, the information is going to be projected along the AP axis. And this is demonstrated by this coronal projection of our 3D venogram shown here. So if we now slightly rotate our view and look at an axial one, if we actually acquire an imaging slice that is on this plane, we know that different veins will drain into our imaging slice at different positions. So here are 4 such example positions. And in our next experiment, we're going to show that by imaging an axial slice, and then by just selecting different regions from it, we can effectively resolve a 2.5D displacement spectrum. And we call this approach 2.5D since it provides an additional selection due to the geometry of the venous system. So for our second experiment, we place an axial imaging slice that's towards the top of the head. And for each image voxel, we acquire a 2-dimensional coronal displacement spectrum. The scan time for this sequence was 20 minutes. And in order to visualize our results, we're going to select different ROIs from our imaging slice and look at their displacement spectra separately. So here's the first ROI that we select, which is on the posterior section of our axial slice. And next, we show an axial projection of our venogram. And here's that selected ROI once again. If we slightly rotate our venogram, we can actually visualize the vein that we expect to drain into this ROI. And here's the coronal projection of that section showing that same vein. Then we can overlay the displacement spectra of this ROI on the coronal projection of the corresponding section across multiple mixing times to obtain the following. We then select 2 new ROIs from our imaging slice, and we expect that the blood will drain into these ROIs from the 2 veins shown in the corresponding colors. We can once again overlay the displacement spectra of the 2 ROIs on the coronal projection of the corresponding section. And now we can show that although we do not perform any displacement encoding along the AT axis, just by simply selecting different ROIs, we can also resolve information along this axis. So overall in this work, we've shown the potential of DiSpect to be used to study the cerebral venous system. Some downsides of our method are that it requires many encodings, resulting on a longer scan time on the order of tens of minutes. Currently, we're working on ways to reduce the scan time. And in addition, we currently do not have any quantitative analysis and verification of our technique. We're working on developing quantitative models in order to assess our technique. And the main advantage of DiSpect is that it can really provide very, very rich information on structure, flow, and perfusion. And we believe that one potential application of this technique could be to be used to study arteriovenous malformations where arteries and veins form very complex geometries. Yeah, thank you so much for listening to the presentation.